Hello my dear students, in this video now we are going to study about the flash photolysis. Flash photolysis is a probe technique. Probe means a search. So by using this technique we will study the rates of very fast reactions which are completed in the fraction of seconds. That means in less than 1 minute time. Okay, and by using this technique we will study the lifespan of the free radicals. Free radicals are very active species and their lifespan is very less. Their lifespan generally lies in the range of the microseconds only. Here in this flash photolysis technique, we use the two types of light sources which are perpendicular to one another. This is one light source and this is one light source and this is another light source. So these two are lying perpendicular to each other. So one light source is here known as the spectroscopic flash or monitoring flash. This is also known as the probe flash. Probe means search. Okay, and this light emitted here is known as the photolysing flash or excitation flash. So here this flash is used to conduct the excitation or the photochemical reaction, and this light source is used or this is used to estimate the absorbance. Okay, so that's why it is known as the monitoring flash. Okay, and here we have to know one more thing, that this what is that photolysing flash or excitation flash is created by using very high voltage current. Approximately 1000 voltage current is used to produce this excitation flash. Okay, and the energy of this flash lies about the 10 to the power of 5 joules. So this much energy is produced by this flash, and this flash exists only for 10 microseconds. 10 microseconds only. So a lot of energy is supplied to the sample chamber or reaction vessel for very less time. Okay na? So short time, for a short time high amount of energy is supplied to the substrate. So when such amount of large energy is supplied to the substrate for very less time, that means 10 microseconds, what happens? The reactant substrate molecules get energy and go into the excited state here. So we can write like this. So m is the what is it? Assume that m is the substrate. M is the reactant molecule. So when it gets the energy from the flash, excited flash or photolysing flash, it go into the excited state. This ground state molecule observe radiation of one wavelength. For example, assume it has the lambda 1, and the excited molecules get observe the radiation of the another wavelength. For example, assume it is the lambda 2. So by measuring the absorbance of the lambda 1 and lambda 2, we can estimate the concentration of the otherwise we can observe the changes that occur in the concentration of the M and M star molecules. Yes, for example, here the reaction between hydroxyl free radical and SO2 that occurs in the environment is a second order reaction, and it is a very fast reaction, and the lifetime of this reaction is lies in microseconds. So speed of such fast reaction is studied by using the flash photolysis technique. Actually, it is a second order reaction, and the rate depends on the concentration of the hydroxyl radical and the sulfur dioxide molecules. So here we carry out this reaction in the lab, and here we take the hydrogen peroxide and mix with the sulfur dioxide. But actually here in the lab we made it into the first order reaction by taking the sulfur dioxide in large amounts. So when we take the sulfur dioxide in large amounts, the rate becomes like this. Okay, so it becomes the pseudo first order reaction. Okay, but anyhow, when we throw the flash on the hydrogen peroxide, this hydrogen peroxide decomposes and forms the hydroxyl free radical. So in okay, within small time large amount of hydroxyl ions are created. So that's why we observe a steep increase in the concentration of the hydroxyl free radical. So here look at this, indicates the flash photolysis technique. Look here. So by using the flash photolysis technique, in a microseconds of time, we produce a large amount of the activated species. Okay. Then afterwards, what happens? This hydroxyl free radical parts reacts with the sulfur dioxide molecule according to this reaction, and changes into the bisulfite free radical like this. So as the concentration of the hydroxyl free radical decreases, the graph goes down like this. So as the time passes, the concentration of the hydroxyl free radical decreases. Okay, so this is due to the flash photolysis, and this is due to the reaction with the sulfur dioxide gas. Here time to time we measure the concentration of the hydroxyl free radical, and we draw the graph between LNOH free radical and time. So we get this type of graph, and we can estimate the K prime, that means the rate constant for the pseudo first order reaction. And by using this pseudo first order reaction, we can calculate the rate constant for the second order reaction, that is K, by using this relation. Already we have learned that the reactant molecules in the ground state observe one wavelength, and they observe the another wavelength in the excited state. No? For example, assume that the wavelength of the radiation observed by the ground state molecule is lambda 1, and wavelength of the radiation observed by the excited molecule is lambda 2. So due to the flash photolysis, what happens? The rate concentration of the reactant molecule decreases. So as a result, the absorbance of the lambda 1 radiation decreases as the time passes, and as the concentration of the excited molecule increases, the absorbance of the lambda 2 radiation increases. So this also helps in understanding the or in observing the changes in the concentration of the molecules in the ground and excited states. For example, here we can take benzophenone as the best example for this one. Here benzophenone is dissolved in the methylized solution by using a TAB. Here when it is subjected to the laser beam, the benzophenone observes the wavelength of the 355 nanometer, and first of all go into the exit state, and then after it goes into the triplet state. In the triplet state, it abstracts the hydrogen from the methyl and changes into the benzophenone ketyl free radical, and this benzophenone ketyl free radical observes the 550 nanometer laser beam. So here look here, in the ground state, this benzophenone observes the 355 nanometer wavelength radiation, whereas in the excited state it observes the 550 nm wavelength radiation. So here in the ground state and in the exit state, different types of radiations are being absorbed. So by measuring the absorbance of these two types of the wavelengths, we can we can observe the changes in the concentration of the molecules in the excited and in the ground states. So that is the use of the flash photolysis technique. Thank you. And this is nothing but